Hi, my name is Alanna Sowles. I'm a University of Oregon neuroscience major, and today I'm going to talk to you about my project with the Summer Undergraduate Research Fellowship, investigating novel psychoplastogenic mechanisms with the help of my friend right here, the fruit fly. Now, after hearing that, you may be asking psychoplastogens and why use fruit flies in neuroscience research, and why does this research matter in the first place? So to answer that, I'm going to start with some background on psychedelics in neuroscience research. Growing clinical evidence suggests that psychedelics are effective in treating intractable psychiatric diseases like PTSD, substance use disorder, and depression. These studies show that psychedelic compounds have valuable therapeutic potential, but the mechanisms of action underlying these effects remain unclear. We're finding that psychedelics may reverse the disease process by acting as psychoplastogens, or substances that induce neuronal growth, for example, dendritogenesis or synaptogenesis. And this is very helpful because our target illnesses are marked by cortical neuron atrophy and dysfunction. And though these psychoplastogenic mechanisms are thought to be independent of the hallucinogenic or subjective activity of these substances, there are limited pharmacological studies investigating these mechanisms. Which brings us to our friend Drosophila melanogaster. Fruit flies are a well-characterized, genetically tractable invertebrate model organism, very popular in the neuroscience field because they have short generation times, stereotypic neuronal morphology, and easily quantified locomotor behavior to establish phenotypes. And though humans are admittedly very different from fruit flies, they do exhibit strong evolutionary conservation, meaning their neurons are genetically, morphologically, and functionally similar to those of mammals. Recent findings show that serotonergic psychedelics like LSD and DOI promote structural and functional plasticity in various model organisms, but there are few published studies utilizing Drosophila in relation to psychedelic pharmacology. And due to their value as a model organism, as we just discussed, expanded Drosophila research in this field would likely complement related studies in mammals. According to a 2018 publication by Lee et al., Drosophila class 1 mechanoreceptor neurons exhibit psychoplasticity in response to the psychedelics LSD and DOI. Both drugs were shown to increase the number of dendritic branches in larval neurons, but much remains unknown about the modes of action at the molecular level. Now, considering this region of interest, class 1 dendritic arborization, or DA neurons, are very well characterized with stereotypic morphological paradigms. The epithelial location of these three class 1 neurons makes for easy imaging, and they have expansive published precedents for baseline reference. So these class 1 DA neurons are located in the peripheral nervous system, and they are segmentally repeated throughout the larval body. Here is a diagram showing the entire dendritic arborization system, which is separated into four separate classes of neurons. Our region of interest are class 1 neurons, and as you can see, they're located in the epithelium or close to the body surface. So here are the leading precedents from the Lee et al. Paper. As mentioned before, their results show that LSD and DOI cause a significant increase in the number of branches in early and late exposed developing larval neurons. So far, my project has focused on replicating these early exposure experiments. So now that we have that background, my research asks two core questions. Can we determine the psychoplastogenic properties of serotonergic psychedelics in developing neurons using Drosophila melanogaster as a model organism? And can we demonstrate psychoplastogenic effects on significantly shorter timescales than published expectations? The ultimate goal of this research is to accelerate mode of action studies in Drosophila larvae by improving experimental design, and we have three aims to accomplish this. We're starting with replicating published precedents, and then we'll move on to optimize drug exposure protocols, and finally, quantify in vivo changes in dendritic morphology in developing class 1 neurons. Our central hypothesis is that serotonergic psychedelics will produce significant increases in the rate, area, and complexity of dendrite growth in developing class 1 DA neurons. Much of this research is uncharted territory, and with limited precedents available, I had to face a lot of unknowns in constructing the methods to pursue these questions. And so much of the summer was spent assembling and troubleshooting my experimental protocols based on extensive literature reviews. Since these early experiments aim to replicate the findings published by Lee et al., our overall design was based on their method. I used a transgenic fly line that selectively drives RFP, or red fluorescent protein expression, in class 1 neurons. And following their dosage and exposure protocol, the DOI or control substance was dissolved in yeast paste to be fed to the larvae. Once hatched, the larvae undergo chronic exposure to either the DOI or the control yeast paste and then are imaged at about 72 hours old. Now, one limitation of the Lee et al. Paper is they had a sample size of 3 larvae, and they only looked at one of the 3 class 1 neurons. Since our main goal is to improve experimental design, as you can see here, we decided to take things a couple of steps further. My replication experiments are comparing treatment groups in all 3 class 1 neurons in 3 different segments on the left and right side. Ideally, this will lend greater power to our statistical analysis and aid in characterizing the full class 1 system. And mirroring the Lee et al. Precedents we hope to replicate, the hypothesis for these early experiments is that dendrite length and branching density of developing class 1 DA neurons will significantly increase as a result of chronic DOI exposure. So now I'm going to walk you through some fun pictures of the hands-on bench work associated with these experiments. Here on the left is an example of how we collect the flies and their progeny and then separate and assemble them for drug exposure. On the right is an example of this yeast paste drug exposure protocol. From hatching to L3 at 72 hours, every 24 hours, the larvae are transferred to fresh yeast paste caps treated with either DOI or the control. And as you can see, the yeast paste is spread across the entire surface of the agarose cap so that the larvae have no choice but to be exposed to the substance and to eat the paste. Now, before imaging, we have to anesthetize the larvae in order to get the clearest pictures of these neurons possible. After combing through the literature, we found a lovely precedent by Kakanja et al., where they use a diethyl ether anesthetization chamber. Here on the left are their instructions, and on the right is my application of those instructions. A really fun part was constructing these tiny little larval chambers out of Eppendorf tubes and very fine mesh. To photograph these neurons, we use a Nikon spinning disc confocal microscope with a 40 times magnification objective. And essentially, the lasers in this microscope interact with the red fluorescent protein being expressed in our neurons, and that helps them to glow, helps us to visualize their morphology. So we've treated our larvae, we've prepped them for imaging, and now it's time to show you the pretty pictures we took of the 3D neuronal structures. Despite our admittedly ambitious sampling goals, we did manage to get a sample size of about 7 neurons in each treatment group for all 3 neurons. Imaging these early replication experiments yielded about 225 total neurons for me to now create 3D reconstructions of in a program called Imaris. Here's a photo of one of our neurons, DD8D, from the control group. And as you can see, these neurons are known for their comb-like stereotypic morphology. And here we see the neurons DDAD and DD8E from the DOI treatment group. And using these 3D confocal images we've collected of the neurons, I then go in Imaris and create 3D reconstructions of each little detail of these arbors. This is an example of our third neuron, VPDA, from the control group. And here again, we see this nice, simple, standard comb-like shape in the arbor. So I take this and construct this filament skeleton from the image. Here's another example of VPDA from the DOI group, and here is my 3D reconstruction. I can then use these structures to extract values and measurements that help us quantify the morphological features of these distinct neurons. So far, I've reconstructed 75 of the 225 neurons in this dataset. For now, these metrics will guide our statistical analysis in comparing any significant differences in morphological features between treatment groups in all 3 of these class 1 neurons. So to wrap this up, this project is still in progress, and as of now, our primary goal is to complete satisfactory replication of the Lee et al. Findings. And pending successful overall replication, we'll proceed to in vivo time-lapse imaging experiments. Now that we have access, we also hope to explore additional psychoplastogens such as LSD and psilocybin. Ultimately, we hope that this research will aid the scientific community in creating safer, more effective treatments for intractable psychiatric illnesses. This project is an ongoing collaborative effort between the Christo and Sean Lockery laboratories with the Institute of Neuroscience, and I'd like to express my sincere gratitude for the opportunity to conduct this research. Thank you for watching. I hope you enjoyed this presentation, and I hope you learned more about the value of psychedelics in neuroscience research. Take care.